Melanoma Matters is an educational resource. The content represents our opinions and does not constitute medical advice. If you're a patient with questions specific to your situation, please consult your medical practitioner. Hi everybody, welcome to Melanoma Matters. I'm James Larkin.
And I'm Sapna Patel.
Today we're going to be continuing our discussion of injectables, and we're going to talk a little bit about SD101. But before that, a couple of quick questions for you, Sapna. So first of all, what is your most used emoji?
Oh, I'm not a big emoji person. It's going to be probably a heart or some version of hearts, I guess.
Okay, so another one then in that case. If you were a wrestler, what would be your entrance theme song?
Oh my gosh. That's not one I even have an answer for. Do you have an answer for that question?
No, I haven't really thought of it before.
Me neither. I've never thought about that.
So maybe we can save that for the next one in that case. No, it's
All
kind of
right.
an interesting question. Okay, so we talked a little bit about TVEC already, and actually it's a very interesting area, this injectable area, right? And there's been a lot of activity, I guess, over the last several years in this space. So the natural thing to talk about, I think, is SD101, a bit of data here. And I guess the main thing we're going to be thinking about is this paper from Cancer Discovery. Tony Rivas is the first author. This is October 2018, SD101 in combination with pembrolizumab in advanced melanoma. So maybe start by thinking a bit about the background in the trial and what it's taught us.
Sure. And let me give a shout out to Teresa Medina, who's the second author on this study. This is a study of a different intralesional from TVEC. We've been talking about TVEC, or, so far that's the intralesional you and I have discussed. That's an herpes oncolytic virus, so viral oncolytic therapy. It takes an attenuated virus and stimulates the immune system. I lovingly called it herpes light, which some people don't like that. This is toll-like receptor 9 agonism. So SD101 is a class C toll-like receptor 9 agonist. Toll-like receptors are comprised of these CpG oligonucleotides, which actually essentially work in a pro-immune stimulatory fashion. Maybe I'll just leave it at that for the very brief description. So it is not stimulating the immune system because of inducing, a mild infection or because of any viral proteins that might be immune stimulatory. In fact, it's just working on natural immune signaling via toll-like receptor 9. So it's an intralesional injection of that. SD101 is the name of that toll-like receptor 9. And then it is in combination with pembrolizumab. So, it might make you ask, was there data on SD101 as a monotherapy? Actually, I have no idea. We can pin that for the fact check because I really don't know. We're going straight to combination here because that's what we're interested in. This was a phase 1b study. So like most phase 1 studies, it was tasked with safety and tolerability and perhaps determining the RP2D recommended phase 2 dose. The study it was a very small study, 22 patients. It had 9 patients who were treatment naive, and then 13 patients who had been exposed to PD-1 therapy. So perhaps I should say PD-1 naive. Maybe they weren't fully treatment naive. And because the primary endpoint was safety and tolerability, the main point, I think, is just to describe maybe the schedule, and then we can talk about safety and tolerability. Short answer, yes, it's safe and tolerable. The schedule of SD101 is essentially weekly, and they gave it weekly for 4 doses. So weeks 1, 8, 15, and 22, and then every 3 weeks for the next 7 doses. So that takes you out, several halfway through the year, if you will. I guess the important thing, safety and tolerable, yes, maybe we can go into more detail if we'd like to. But as far as efficacy, the treatment naive or PD-1 naive population had a really strong objective response rate. 7 patients were evaluable. If you look at the figure, the spider plot, or spaghetti plot, as some people say, it's figure 1A. This is the PD-1 naive population. You see that the kinetics of all of those 7 patients are downward. So there seems to be objective responses in all of those 7 patients, but it was 9 patients, 1 patient who had rapid progression of disease, and then another patient who developed pneumonitis and was unevaluable. So if you do the 7 out of 9 objective response rate, then you get 78%. And in the PD-1 experienced population, it was an objective response rate of 15%. So 2 of those 13, both of those were partial responses. It's probably worth noting a couple of the PD-1 naive patients were actually complete responders. So really interesting here that you can see with this toll-like receptor 9 in a PD-1 naive population a much higher response rate. Maybe that argues that that's the pembrolizumab doing all the work there, though 78% is higher than we would normally expect in frontline therapy. Although this is all patient selection, right? I mean, when you get the right patient population, you can get extremely high responses to single agent PD-1, and it is just 7 patients. So massive caveat there. And I guess the second thought has a bit escaped me. Oh, the second point to make. This study was really rich on correlative. So what they did a really nice job at was doing on-treatment tumor biopsies. And the short answer is they basically saw an influx of CD8 T cells and an upregulation of interferon gamma in these injected tumors, and preferentially in the responders. So if you were a responder, you were more likely to have upregulation of RNA, PD-L1 by RNA expression profiling, as well as interferon gamma and an influx of CD8 T cells. But you also saw that even in the PD-1 refractory, where there wasn't as many responses. So that's the takeaway.
Yeah, and I think it's really interesting. I mean, I'm sort of thinking about it in the light of Master Key 265 in some respects, and the phase 1B really. In other words, however many, 20 patients, something like that, then response rate around about 60%, bit more, jump straight into a phase 3. And then here, I'm seeing this really very high response rate in a smaller group. And I'm just thinking to myself, well, maybe the right thing to do would have been to seen in a few more patients what this looked like, 20, 30, 40, whatever, maybe even think about randomized phase 2 as a basis for further development. That's one thing. And then the other thing is the kind of efficacy in the refractory setting, right? So maybe just a couple of quick words about this now. We haven't talked too much about this, but it's an important subject. What do you think is a worthwhile level of efficacy in the refractory setting, and how would you define that efficacy as well? So 2 separate things, really.
Yeah, and I think this study was trying to get at that. So, I think most of us pre-approval of lifileucel, Amtagvi as it's branded in the US, TIL therapy from Iovance. Prior to that, I think most of us would have said in a PD-1 refractory population, probably 15% is our bar, unless you're doing dual checkpoint blockade. SWOG S1616 studies showed you could maybe get a 27% response rate in PD-1 exposed patients who've not had a response. But so we're around about 15 to maybe 30% objective response rate. Ideally, you want that to be somewhat durable, but we have very limited options in this setting. I think what you're also asking is, this study was trying to do that thing that I've heard some people sort of turn their nose at the phrase turning cold tumors hot. They don't like that's very simplistic, but I think it does help frame to people what's happening. You have a PD-1 experienced patient. That means those tumors have seen anti-PD-1 therapy, but for whatever reason, it's not responding or it's lost its response. So what does that mean? It means either the T cells have stopped trafficking to that tumor, PD-L1 is no longer being expressed in those tumors, interferon gamma is not being secreted to induce the PD-L1. So something about that tumor microenvironment has cooled off in some way. It either was cold to begin with or it has cooled off. And by injecting into that tumor even if the injection itself won't drive a long-lasting response, you're basically trying to turn that tumor hot so that the immune system recognizes it and traffics it. That was essentially some of what was happening in that PD-1 refractory, turning a cold tumor hot. And I think they proved that you can do it. That was that's the reason of such a strong publication impact factor here, not just that it has Tony Rebus's name on it, but that he did real good science here, right? He looked at the tumors and said, we are turning some of these cold tumors hot. It may not be sufficient to generate a resist response, but we are augmenting that biology. Does that sort of maybe get to what you were saying?
Yeah, and then I suppose the next practical question, in as much as you know about this, people might look at these data and think, oh, what happened? What's happened to SD101? Why haven't we seen more of it? Is it because people weren't that impressed with the data, or is it more of a pragmatic type of consideration in terms of where the drug is, I guess?
Yeah, I mean, you bring up a good point. Should this study have lent itself to some sort of intermediate size, maybe even randomized, just to explore more this signal of benefit? My knowledge of this topic, extends to just what happened to the manufacturing and the development of SD101. It was sold from Dynavax to another company, to Trisalis. And, Trisalis really has as a mission to go after rare tumors and cancers that affect the liver, in which case intralesional may not be the approach that really suits that visceral setting. And they have gone on to develop SD101 as more of an intra-arterial strategy, which, by the way, most of these toll-like receptor 9s are intralesional. There are others that we can talk about, but most of them are intralesional. So to put a toll-like receptor 9 directly into the arterial blood system, isn't without caution. But Trisalis did plenty of animal studies, preclinical studies, to show that it should be okay to be able to do this. The concern would be that you would have roaring cytokine release once you just start activating the immune system. And it turns out, there's some data we can talk about maybe at some point. This intraarterial approach in the liver really did not demonstrate any safety signals associated with cytokine release in a maximum tolerated dose or dose limiting toxicity way, I guess I should say. So it is safe to do that. I think the development now has just moved away from cutaneous melanoma and intralesional for this reason. The drug has been acquired by another company who has just a different development plan for it. But, for other toll-like receptor 9 agents in this space, you wonder if there is some room for development in this PD-1 refractory space. And there's a couple of other trials we can eventually talk about.
Yeah, so that sort of maybe brings us to the final thing I wanted to ask anyway, and I don't know if this is an elephant in the room or not, but we know pretty well the Illuminate 301 trial, the randomized trial with toll-like receptor 9 agonist, presented a couple of years ago, I think now, or more maybe. It is negative. I don't think we've seen publication yet, but I think we're going to get an update on that at ASCO, so we're looking forward to seeing that. So, big question, these drugs all the same as each other or not? Because it might be, just how do you interpret this in the light of a negative randomized trial that hasn't been published yet? What do you think?
Yeah, I don't think they're all the same, and maybe that's just me saying what all the companies would want you to say, which is don't compare ours to the other. They're not the same. But SD101 is a class C toll-like receptor 9 agonist. There are class B agonists, which is, like what you're alluding to. And they have different activities for how they can stimulate, say, the immune system and how they might eliminate MDSCs. That's part of how, toll-like receptor 9 might work, is it might also just change the balance of effector and regulatory and cells such as MDSCs. I don't think they're all the same, but we see this in multiple places. We've also seen it in the 4-1BB agonistic space, that there are different 4-1BB agonists that just, for whatever reason are not the same, depending on whether they have certain antibody features, built into their drug. So I don't know. My short answer is this is not a Nivo pembro situation where we think of Nivo pembro as fairly interchangeable. I don't think we're going to be able to necessarily interchange all these toll-like receptor 9s.
Yeah, no, and I think that's a really important point. Sometimes it is right to think about things like Nivo pembro. Other times, actually, it's important to be aware that isn't correct, because just it kind of feels to me that there might be a place, for these agents in therapy. There's a signal there, and I guess the question is, how do the drugs get developed, and what's the right place to try and look for that signal? Ideally, obviously, in a rational way, if we can. Okay, so anything else that we need to say about that, or do we have a wrap, do you think?
I think we're good. SD101 plus pembro, high, safe and tolerable, and high response rate in PD-1 naive, less so in PD-1 refractory, but the tumor biology is there. It's drawing in T cells and upregulating interferon gamma.
Okay, thank you very much.
Thank you.